of disparate genomic resources using Galaxy. I'm a member of the Galaxy team. So I was originally going to be skipping the what is Galaxy portion of this talk, but after hearing some of the talks today, definitely need to reiterate what Galaxy is. So what is Galaxy? Galaxy is a data integration and analysis platform. It enables reproducibility, accessibility, and transparency. Users without programming experience are able to easily specify the parameters to run tools and workflows. It's reproducible because Galaxy captures all the information that a user does when they perform their analysis. There's no need to record or perform any extra steps to do this. This is all automatic when you use the interface. It's transparent in that it allows users to share their work and to publish their analyses via the web using interactive web documents known as pages. So first and foremost, it's a data analysis and integration tool. It's a free for all. That's everyone, commercial, academic, anyone is free to use our web service. We integrate a wealth of tools, a large number of compute resources, terabytes of reference data, along with permanent storage. I want to point out that it works on all modern web browsers, not just Chrome. It's open source software. It makes integrating your own tools and data and customizing it for your own site very easy. There are, in fact, several ways to use Galaxy. We provide a free site at usegalaxy.org. If you register a user account, you get 250 gigabytes of free compute. And you're more than welcome. If you exceed that amount, you have larger amounts that you need to use. You don't want to wait in the public queues. You can go ahead and you can download Galaxy from getgalaxy.org, and you can implement it on your own hardware, have it run on your existing clusters. It's also very easy to launch Galaxy in the cloud. You can use Cloud Launch at launch.usegalaxy.org. This uses the Cloudman interface, and we heard quite a bit about that earlier today. It's very straightforward to use Galaxy in the cloud in AWS. You sign up at Amazon, you give them your credit card information, you get your secret key and your passes, and then you just use this. At no point does the Galaxy team or the Galaxy project or anyone get any of this billing information. It's totally hands-off. Now, we realize that our instance at usegalaxy.org, it cannot fit all the needs of all different kinds of domains. We do focus on next-generation sequencing and analyses on that. But there's a huge number of different public servers that have been made available by other projects and other groups. And that number is at 60 plus public servers. And hopefully that number keeps going up. In Galaxy, when we talk about various things, we have this idea of a data set. This would be any input or an output of a tool or the intermediate steps of that data along with the associated metadata. It's basically a record of a specific analysis step. And these are immutable objects, which means once you create them, you can't go back and change them. A history is a series of inputs or analysis steps and along with their outputs. And so as you perform an analysis interactively, you build up this history. I'll show a screenshot very shortly. And this is a record of the analysis path that you did with your data. A tool within Galaxy is any operation that acts upon a data set or creates a data set. And these can be developed by the Galaxy team or the community, or it might be a third-party program that's been wrapped into Galaxy, such as BWA Bowtie, for example. A workflow would be a series of analysis steps executed as a unit. When we talk about sharing something inside of Galaxy, that means that you've made it available to someone else. You can share objects either directly by their email address or by creating a link that you can share. You can post on web pages, you can send in emails as well. When we talk about publishing something in Galaxy, this means that we've made it available to everyone. There's a special section at the top of the masthead inside of Galaxy called Shared Data. And here you want to make your Galaxy objects available to everyone. They'll be listed in a public place. People can go and get copies of them, look at them. Once they have their own copy, they can modify them as they wish without you having to worry about them interfering with your original analyses. Galaxy page inside of Galaxy. This is an analysis document. It's very much just like using a word processor. except it's on the web. It's what you see is what you get, but you can embed Galaxy objects such as histories, workflows, and visualizations. When we talk about visualizing inside a Galaxy, we talk about sending our data to external resources or using the built-in Galaxy Trackster genome browser or using Galaxy charts or their visualization plugins. This is the Galaxy interface. You can see at the top we have our masthead. Clicking on these links, if we're in the Analyze Data interface, which is what we're in right now, you can see on the left-hand side here, we have our tools menu. If you click on a link here, it'll expand that tool section to give you the tools that are available within that link. Clicking on a tool will put that tool into your middle pane here. And then here you're able to configure your tool, select your input data sets, configure parameters such as in this case we're doing a BWA alignment. So we select a reference genome, whether or not it's paired end or single end, what settings we would like to do when we perform this, and then we can click execute. Clicking execute will put a new data set into your history here on the right-hand side. It starts down at the bottom at 1 and works its way up. Each time you run a tool, it will create one or more data sets in your history, and you can then use those data sets to perform additional analysis steps. There's several different tool states that a tool can end up in. Green is always good. You want your data sets to turn green. This yellow, tan, it means that tool is actually running. It's being computed right now. If a job is queued, either because the cluster is full, or in this case, step 7 here relies on step 6, it will be grayed. If you actually have an error, it will turn red. If you find errors inside a Galaxy, you can expand that tool section, and there'll be a little bug link. You can click on that bug link to find out more information about it and to report the bugs to the Galaxy team. Please do report bugs so that we can fix them if you find any. In addition, you have several other options here inside of expanded tools. So you just click on that dataset title there. It'll expand the dataset. You can click that rerun button. What that will do is actually pre-populate the center pane with the tool that was used to create this dataset, along with all the original parameters that were selected. You can, of course, click this floppy disk icon to download. I was talking to someone before. They said that was a TV, and they had no idea what a floppy disk was. So that is a floppy disk for anyone that doesn't know. You can additionally click on this eye icon and find out all the information about that individual tool run. So in this case, we don't have a black box. We know exactly what happened. We know, in fact, the Galaxy tool version, the version of the Galaxy's own internal tool version that was run, along with the actual binary that was run on disk, the entire binary version along with that actual tool itself. We have UUIDs that are created, and we actually, depending upon the configuration of your cluster or your Galaxy instance, you can actually see operating system, the full command line, the amount of runtime, cores that were allocated, and so forth, and along with a list of all the parameters that you had selected before. So no black boxes here. In addition to tool pages, so one of the things we try to do inside of Galaxy is make it very easy to run tools. And so we provide a commonly used set of default parameters when you first when you first load a tool interface. So here we can see that we have commonly used selected, and then the actual tool author here of this is the last Z, which is a mapping tool for longer reads, has actually predefined several different assumed percent identities for your alignments here. However, you can always go ahead and click on full parameter list and get the entire set of parameters. And so you're not limited to some minor subset of parameters. You can always get the entire parameter list for that tool, depending upon the way that the tool is wrapped by that individual author. Okay, so tool suites inside of Galaxy, we have lots of tools available. We have a Galaxy Toolshed, which is an app store for Galaxy to install tools. And so the Galaxy team itself can't wrap all the tools. We try to have community resources available. So we provide visual analytics directly within inside of Galaxy. This is Trackster browser. We can actually rerun tools on individual regions or across the whole file set. And we can reconfigure directly within this genome browser, run it, reiterate across until we find the parameters that we like. We can also do dynamic filtering by selecting these sliders, sliding back and forth, you'll see these transcripts fade in and out of existence. To create a workflow in Galaxy, you can create it from your interactive analysis history. Just click Extract Workflow. That'll create a workflow that's pre-populated with all those options that you found before, and then you can just go ahead and you can run it. You can also manually edit workflows or create them from scratch within our workflow editor. When you go to publish a page inside of Galaxy, you write a paper, you want to include information about it. We actually have Galaxy Pages. You can include a link directly to that page inside of Galaxy. And then when the user clicks on that link from that paper, they can actually get this nice Word document that was created. And so you can actually have your histories. They can import them if they like. They can view them, share them later on, rerun your analysis all within their web browser. In addition to all those features, we now have interactive environments inside of Galaxy. So you can actually load up by clicking on this visualization button here. You can actually pull up and create an RStudio interface inside of Galaxy. The same thing exists for iPython. If you'd like to know more about this, see Bjorn Gruning. This is the Galaxy team. Several of us are here. If you have questions, please ask them and buy them a beer. We have a large number of contributors and extended members of the team, 50 plus contributors. In fact, I only was able to provide pictures for people that had 100 or more commits to the Galaxy code base. Okay, so now onto the real part of the presentation. So to get Galaxy data into Galaxy, you can either upload from your computer existing URLs, FTP for large files. That's pretty boring. But if you want to get data from existing genome resources, we provide direct interactions into them. So for example, Genome Space, the UCSC table browser, and Biomart Intermine, for example. The way this works with Galaxy is it uses a callback URL. So the user would start at Galaxy. They would click on a data source tool. They would then be forwarded to that data resource within their web browser at that resource's location. And we send with the user a Galaxy URL value that tells them where to send back that data. The user interacts normally with that external resource, and when it's time for them to retrieve data from Galaxy, they click that Send Data to Galaxy button, Export Data to Galaxy button, however the external resource decides to phrase it. And then that will do a post back to our Galaxy server, and that will be the Galaxy server specified by that Galaxy URL, so this will work with any Galaxy throughout the world. And it provides us with a new URL value and parameters needed in order to retrieve that data. The user is then forwarded back into Galaxy, and in the background, a job is launched that retrieves data at that URL from those provided parameters. And here we have a set of genes that were retrieved from UCSC. We also provide an asynchronous workflow. The user clicks the same way, except when the user gets sent back to Galaxy. In this case, that site has a long-running process required to generate that data. So Galaxy instead submits the parameters to the URL along with a new Galaxy URL that would specify this data set. When that job's done running at that external site, that external site will post back to Galaxy URL with that data. To add your own, you just use an XML file. I don't have a problem with XML. I like it. It works well. You just specify that the external site that you want to access. This is yeastmine, and then you tell Galaxy to load that tool file. So last year at the GCC 2014 hackathon, and then it continued into the BOSC hackathon, Matt Shirley and myself, we worked on a generic approach to get multiple data sets along with their metadata sent back to Galaxy. And so you can read all about this process that we have assembled, along with links to a tool in the tool shed that will actually implement this process. That's all available at our Trello board. You can also do a customized approach with Genome Space, where we use OpenID, and this is required to get the username and the temporary token. The tool itself interacts directly with the Genome Space API to fetch the data and metadata. If you want to know more about these, you can check out those files. To get data out of Galaxy, there's two different ways to do it. We have external display applications, where you can send data to Galaxy. You click on a link. In this case, we're sending it to IGV. This is a Java application that will launch on your own computer. That's why you get a scary warning. To define it, more XML. It's not very scary. You click on that link, view at UCSC main. You get sent directly in your web browser to UCSC main. We also have data destination tools. In this case, you're actually inside of Galaxy configuring that tool, and you can have more than one data set if you want to do that, along with specifying additional values. You click execute, data sets get sent to Genome Space, and then you can have an HTML report that you want to view. You can click on that link, and you can download the data directly from Genome Space. Come to Indiana next year for the Galaxy conference in 2006.